Good morning, good afternoon, and good evening, everyone, and welcome to the third event in the Neurophysiology 2023 series titled New Horizons, Gonadotropin-Releasing Hormone and Cognition, featuring Dr. Vincent Prevot, Research Director and Laboratory Head of Lille Neuroscience and Cognition, and INSERM. Today he will present his research on the development and establishment of the gonadotropin-releasing hormone system and the importance of its first postnatal activation. Before we begin, we'd like to acknowledge our partners for this series: the American Physiological Society, the Society for Neuroscience, the Federation of European Neuroscience Societies, the American Autonomic Society, and Scientist.com. So thank you to all of them for making this series possible. Today's event has been sponsored by PrimeTech, who are makers of the iPrecio micro infusion pump, and so a big thank you to them for helping make this webinar possible. And I'd now like to welcome Chung Tan from PrimeTech to say a few brief words. Take it away, Chung, whenever you're ready.
Okay, thank you, Andrew. So welcome everybody, and thank you for joining us today. So iPrecio micro-infusion pump. For us, it's the only way to deliver compound without interfering at the moment of delivery, and we hope it plays a part in helping you do science. Our mission really is to make it as easy as possible to evaluate molecules in vivo, and of course make the results as relevant to the clinic, so whether it's exposure profile or natural dose patterns, etc. So this is really important, I believe, for you. And how do we make this possible? Well, easy to use application software. You probably cannot see everything here, but just to explain, for example, column 1 would be the flow rate that you would decide to program the pump on, and then duration is the second column. Basically, you will decide if you just want one step with this continuous infusion, or you can do multiple steps. On the left-hand side here is just to show you what is possible. You can program repeat cycles on 24-hour cycles or weekly cycles. So we feel it's quite easy to use software, and you can reuse it in the future. What does the pump look like? We said make it as easy as possible to use. So we need the device to be small, easily implantable, and then you can actually refill it through the skin as well without any additional surgery. So I'm going to play a video now, and hope you'll be able to see what iPrecio pumps are about.
iPrecio is equipped with a very small battery-operated micromotor which rotates anti-clockwise at a very slow speed. An elastic tube that contains the infusion solution extends from the reservoir to the exit point of the pump. The shaft of the pump motor is equipped with a rotating cam. As the cam rotates in an anti-clockwise direction, 7 pins are pushed outward in order to place pressure on the elastic tube. This pressure is applied in a uniform sequential manner, allowing fluid to be pushed through the tubing in one direction. The fluid in the elastic tube is displaced to the left very slowly by this unique method of peristalsis. Consequently, the fluid goes out of the pump and is infused into the animal. The rotation of the stepping motor is very accurate and precise because it is controlled by a crystal oscillator. Thus, iPrecio is able to continuously inject microliter quantities of test compound.
So coming back to the slides now, you can see here, this is actually the port that you can access through the skin. This is the flexible reservoir that will go concave as it infuses out, so you can check that it's working as well. And this is the catheter that can be to go to IV or sub-Q. We started in March 2007. We improved the product 2 years later. So this is a SMP 200 pump. These are for rats or larger. We came out with a mouse product in March 2015. And then we improved it 4 years later. The pumping mechanism always the same, but we improved the detection and programming speed of the pump itself. Here are some publications, and actually in the resources, you can download a booklet with highlights from peer-reviewed publications. So this one here, you can see it's actually Charles River with GLP validation of the SMP 200 pump. Finally, just to show you we're at PrimeTech. We are based in Japan, and we are the manufacturer of the iPressio pump. Thank you very much. Thanks, Andrew.
Okay, great. Thanks very much for that introduction, Chung. As Chung has mentioned as well, there's a number of resources available in the handouts if you'd like to learn more about any of those things. Vincent, you feel free to take it away whenever you're ready.
So do you see my screen?
We do, yes. Perfect. Thank you.
Perfect. So here's the pointer. So thank you very much, Andrew, for your introduction, and thank for inviting me to present our work in front of all of you. It's a great honor. So as Andrew told you, I will speak about the gonadotropin-releasing hormone and its newly discovered role in cognition. So what is GnRH, or the gonadotropin-releasing hormone? In fact, it's the hormone that will control all the reproductive processes in vertebrates. And this hormone is released by a handful of neurons that are called the GnRH neurons, that have the cell body located distributed from the olfactory bulb to the hypothalamus. And these neurons all project to another region of the hypothalamus that is called the median luminous of the hypothalamus, where they will release their neurohormone in capillaries that do not carry the blood-brain barrier. They are fenestrated and enables the macromolecule to be released into the pituitary portal blood, where and in these vessels it will reach the anterior pituitary, where it will regulate the release of gonadotrophins that are LH and FSH that will be released into the general circulation to control gonadal growth, gametogenesis, but also the production of sex steroids. So these neurons are fascinating because they are the ones that control species survival since early vertebrates, including in humans. And these neurons that are so important, and that's why we are here today alive, they are not born in the brain, but in fact they are born in the olfactory plaque in the nose during embryogenesis, and will migrate from nose to brain until birth. And at birth, the system will be in place. But we all know that even if the system is in place at birth, it requires a postnatal maturation, and we all know that it's fully active at the time of puberty. That is the first ovulation in females or the first production of spermatozoids in males. And this postnatal maturation is divided in several steps. And as Andrew was alluding to, one of the key steps during postnatal development is this minipuberty that occurs in all mammals from the first week of life and lasts different lengths, 6 months in humans, 2 weeks in rodents. And in this minipuberty, there will be the first activation of the GnRH neurons, and also operated by a switch in the expression of microRNAs in these neurons that will go and inhibit repressor transcription factors that are repressing the transcription of GnRH to activate for the first time these neurons. And this will lead to the first activation of the gonads that will release gonadal steroids, estrogen or testosterone, that will feed back into the brain, not only to mature the GnRH system, but also to act on all the neurons of the brain that have the androgen or estrogen receptor, and to tell them that they have ended a developmental program and they will need to start another one. And in humans, there is a rare disease, a genetic disease that is called congenital hypogonadotropic hypogonadism, in which these GnRH neurons are never activated, or even in a subclass of these patients that have the Kalman syndrome that associate anosmia, inability to smell, and infertility. We think that it's an alteration of the migration of the neurons from the nose to the brain. But luckily, these patients, when they want to have a baby, even if they have no puberty and no spermatozoid and no ovulation, they can be put under a pump system that delivers the endogenous molecule GnRH at the endogenous rhythm to promote ovulation in women to be able to carry a baby, and the production of spermatozoa in men. So here clearly there is a link between fertility and olfaction, because I told you that this subclass of patients have anosmia. But then the question is whether, and also it's known that some cognitive alterations, such as Alzheimer's disease or Parkinson's disease, one of the first signs of this disease is the loss of the ability to smell. So there is a clear link between olfaction and cognition. And the question was whether there could be a link between fertility and cognition. So why we ask this? It's because thanks to the development to new transplantization techniques, we were able to see the full contingency of GnRH neuronal cell bodies and fibers in the brain. And we realized that, in fact, these GnRH neurons, in addition to project to the areas that are involved in the control of reproduction, they are also projecting in areas such as the hippocampus and the cortex, areas that are involved in cognitive processes. And thanks to the generation of a mouse in which we can visualize all the cells that produce the GnRH receptor, so the receptor for this gonadotropin-releasing hormone, and using again these transpartization approaches, we can see that indeed in this region there are neurons that do express at least the promoter of the GnRH receptor, including in the hippocampus and the cortex. Thus, we wanted to know whether these neurons were playing an important role in the cognitive processes. And for that, to test that, we did a very simple experiment. That is to say, to inject into these hippocampus cortical areas inhibitory dread viruses in mice, and we subjected these mice to a very simple test, dishabituation test for olfaction, for example, or recognition of new objects. And we tested the same mice before the viral injections, 1 month after the viral injections, and injecting them saline that should do nothing to the dread expressing cells. And 1 month after, again, we did the same tests, but this time giving CNO, that is the ligand of the inhibitory dread receptor. And here the results were totally impressive. You see that wild-type mice before or after the injection of virus are behaving, they are able to recognize both new odors and new objects. But then when you do this experiment, when you repeat this experiment, and half an hour before you inject CNO to inhibit these neurons here, hyperpolarize them, then you see that they are totally unable to recognize the new odors or the new object, suggesting indeed that there is a link there could be a link between fertility, at least fertility hormone, and cognition. Now, in what physiopathological condition this could play a role? So we were wondering, and then we came across the Down syndrome. So Down syndrome is characterized by the fact that it's a chromosomic alteration that will cause neurodevelopmental disorders, and these patients have problems learning, and then when they reach a certain age, now because of the progress of medicine, before they were dying very early, around the age of 30, and now they can live very long. But we realized that at the age of 40, almost all of these patients, if they die, they have the neuropathology of Alzheimer. And then when they reach the age of 65, 75% of them develop this dementia, Alzheimer dementia. And interestingly, we have mouse models reproducing or mimicking this disease. And why did we choose trisomy 21 or Down syndrome? Is because on the chromosome 21 in humans and chromosome 16 in mice, you have a set of 5 microRNAs that are thus in 3 sets in these mice, including 4 of them are known to be overexpressed in GnRH neurons, and including the one that is key, plays a key role at minipuberty, that is MIR155. And then, so we went on to study these mice, and we wanted to know whether their disabilities, sensory and cognitive disabilities, were acquired over time or whether they were already there since the beginning. And here what we did is we tested young mice just after weaning, way before puberty, like adolescent mice, let's say, and we test them for odor recognition and object recognition, and both male and females. And here we saw that while at birth the mice leave, so that means that they are sensing the smell of their mother, because to reach the nipple of the mother at birth, they need the required olfactory cues. So at birth, apparently, they are normosmic. But then here we see that in the adolescence, they are already unable to recognize new odors. The trisomic one are in red and the wild type littermates in gray. While interestingly, they still have, at this young age, they still are as efficient in recognizing new objects than their control littermates. However, when we do repeat this test at adult age, we see that they are still impaired for olfaction, but they also begin to be cognitively impaired. They are unable to recognize new objects. So we wanted to know whether GnRH could play a role in this or be associated to this gradual decrease in cognition. And for that, we did just immunoreactivity for GnRH in the brain using our transplantization approaches. And we saw that, for example, we first counted the number of GnRH neurons in the brain, and we saw that they were identical up to adolescence, and then that we had a drop in the number of GnRH neurons expressing GnRH in adulthood. And then our surprise came also when we looked at the transplantized brain. Here is a sagittal view, here is a ventral view. So you see all these GnRH fibers reaching hippocampal area, for example. And here in the Down syndrome mice, you still have GnRH neurons and neurons that project to the areas involved in the control of reproduction. But here you can see that you lose all the immunoreactivity in extra-hypothalamic area, including hippocampal and cortical areas, suggesting that there is a huge decrease in the expression of GnRH in these mice. And assessing hormonal levels, so we can assess the fitness of the system by accessing the proxies that are LH and FSH. And here, surprisingly, we see that these hormonal levels in the circulation are higher than control littermates. However, the testosterone levels are identical. But when we analyze more precisely the rhythm of GnRH release, that you can measure exactly with the rhythm of LH release in the general circulation, because one pulse of GnRH will induce one pulse of LH. You see here in gray the normal pulsatility of a mouse, a wild-type mice. You have one pulse every 2 hours. And here, this also occurs in Down syndrome mice, but look, the basal levels are much more elevated than in controls. And in addition, the peaks are blunted by around half. So the pulsatility exists, but it's altered. The pattern is significantly altered. And so we wanted to know whether this decrease in GnRH expression was somehow linked to the overexpression of miR-155 in these neurons that could interfere with the production of GnRH. And here we were very surprised because we were expecting an upregulation of miR-155 and we got no change or maybe a decrease. But also what was even more surprising is that we saw that all the microRNAs of the miR-200 family that plays a crucial role in the control of the GnRH promoter because it inhibits one of the potent inhibitory transcription factor, that is ZEB1, were downregulated. And when these are downregulated, then you are expecting that ZEB1 would be upregulated. And indeed, it's happening. ZEB1 is upregulated in Down syndrome mice, while GnRH transcript expression is downregulated. And other transcription factors that are activatory transcription factor like OTX2 are themselves downregulated. So suggesting that indeed the GnRH expression is decreased in these animals. So then we said maybe if we re-express in adult animals this miR200 family, then maybe we could have a positive effect on cognition or olfaction in these mice. So what we did is we chose to overexpress miR200B and selectively in the provocative region. So we injected this miR200B here in the provocative region. So you see here in green all the neurons that were infected, and in red you have the GnRH neurons. So you see GnRH neurons that are infected. And here we were very happy to see the selectivity of the infection, because here you see, for example, in the tuberal region of the hypothalamus, that you have no green nowhere in the brain except in the nerve terminals of GnRH neurons that project to the median imminence. And here we saw that this overexpression of the miR200 was indeed overexpressing miR200B, but not changing any of the other microRNAs. And then we repeated the test we were doing before on the same mice, that either we injected a similar virus, but that was not overexpressing miR200, and we saw that there was no change neither on olfaction or cognition. And then here, what was very surprising, after 3 months after the injection of the virus, we saw that we were increasing significantly the ability of mice to recognize new odors or recognize new objects at levels that were quite similar to the wild-type littermates. And this had an effect in the brain to increase the expression of GnRH in the provocative region, suggesting that the GnRH neurons were not dead, but then they were just not expressing anymore GnRH, and also the increase of expression of the activatory transcription factor like OTX2. And doing some RNA sequencing in the hippocampus, so in an area that was not targeted by the miR200 virus, we saw that in Down syndrome mice, you have 72 genes that were upregulated and 19 genes that were downregulated. And when we were injecting the miR200 into the provocative region and we were looking at the changes of these genes in the hippocampus, we saw that we were able to rescue the upregulated genes, so downregulate them or correct their upregulation of 50 different genes that were involved in oligodendrogenesis and myelinization. And then, indeed, it looked like when we did in vivo X-ray physiology that in Down syndrome mice, you had a deficit in conduction, a neuronal conduction between the left and the right hippocampus, and this was rescued after the injections into the preoptic region of the miR200. And then we wanted to know whether we could rescue the system also using cell therapy, because there has been in the 80s fantastic studies showing in mice that are mutated for GnRH that are just infertile. Then if you were injecting chunks of provocative region collected from a newborn mouse in the adult brain of these infertile mice, you were able, the GnRH neurons of this explants were able to colonize the hypothalamus and to restore fertility in these mice. So we used the same approach. This time we collected provocative region containing the GnRH neurons from wild-type mice, and we injected them in adult Down syndrome mice, and then tested the effect on cognition and olfaction 3 months after the graft. And then here we saw again that we were recovering in these Down syndrome mice both sensory cue, their sensory abilities and object recognition abilities. But then here you will tell me that when you inject provocative region, you inject GnRH neurons, but many other cells. So this rescue, is it linked to the GnRH neurons? So to test this very important question, what we did, we repeated the exact same thing in mice that were expressing selectively botulinum toxin to block the exocytosis of the GnRH neurons in GnRH neurons. So botulinum toxin cleaves VAMP1, 2, and 3, thus prevents the ability of the neurons to release their neurohormone. And so we did this similar graft from this botulinum toxin mice expressed in GnRH neurons into the provocative region, into the brain of the Down syndrome mice. And this time we saw that the graft, in contrast to the graft of wild-type explants, were not able to rescue neither olfaction nor cognition in our Down syndrome mice. However, if we were doing an acute injection of GnRH in the same mice 1 month after, then we were able to rescue sensory and cognitive abilities, suggesting here this experiment is key to show that GnRH is the molecule that does the job. And then, but all this microRNA, viral injections, cell therapy, this is interesting but very complicated to translate to humans. So we wonder whether we could use pumps to infuse GnRH and rescue the cognition in our trisomic mice. And to do that, first we did use the classic pump that was available at the time before 2015 in mice, that is the AZ mini pump that infused continuously GnRH. And here we knew that GnRH system was a very sensitive system, because if you overactivate the receptor, always it will internalize and then it will shut down the system. So we did try that in wild-type mice. And indeed, when you do a continuous infusion of GnRH in wild-type mice, you have a very negative effect. You induce the loss of the ability of the wild-type mice to smell new odors, and the loss of the ability of this wild-type mice to recognize new objects. And indeed, as you can see here, when you measure pulsatility in these mice injected with continuous infusion of GnRH, it's called Lutrelef. Lutrelef is the medication used in humans, but it's strictly GnRH. You see that you blunt totally the pulsatile release of GnRH in these mice, showing the desensitization of the thing. And if we do the same continuous infusion in Down syndrome mice, you don't have any further deterioration in these mice. However, if we use these fantastic pumps Tsang just introduced before, that are able to be programmable to phenocopy exactly the wild-type pattern of GnRH release, and you see here the pattern of natural wild-type mice and the pattern that we corrected in red in the Down syndrome mice using this pump. Here you see this identical pattern. After 2 weeks of this pulsatile release, wild-type pulsatile release in Down syndrome mice, you see that you are able to rescue olfaction and cognition at levels of wild-type mice. So this was super impressive. And because this drug is used, as I told you, since many years in humans with infertility, we switched directly to humans thanks to a collaboration with Nili Pitelou at the CHUV in Lausanne in Switzerland. And we asked whether this could be of any use for people with Down syndrome. And here first we analyzed the hormones in these persons, and in blue you have the Down syndrome patients with Down syndrome, in white the control persons. And you see here that was also very impressive and important to us. In fact, our mouse model at the hormonal level, at least, phenocopied perfectly the human patients, because you see that testosterone was not changed like in mice, but both LH and FSH were up, at higher level in the persons with Down syndrome than in control person. So exactly the same phenotype than in mice. And then what we do here is that we implant a pump like the one used for insulin, that we can program so that it gives a pulse every 3 hours, a pulse of 70 nanogram per kilo every 2 hours. And then we receive these patients and we perform a cognitive test, MRI test, blood test before and after 6 months of this regimen. And here it was very impressive to see. I have no time to detail, but here, for example, MOCA test is regrouping several tests, cognitive tests. So we were able to do a pilot open label study on 7 patients. So you see here already that the cognitive performances of the persons involved were very different. Some were very cognitively impaired, and others were very close to the performances of the general population. But interestingly, 6 months of treatment improved by 20-30% the cognitive performances of all the patients. And you see here that the one that was the most close to the general population arrived to reach the cognitive level of the general population. So this was impressive, but we need to be very careful because both the patients and the medical team were knowing that the people were receiving a treatment. So the patients want to show they improve and the medical team wants them to improve. So maybe we need to take that with caution. But what was even more impressive, and the neuroradiologists who analyze the data never saw that in their life, is that within 6 months of treatment, using a resting state functional MRI, we saw that this treatment changed the functional cognitivity in networks that were known to be downregulated in persons with Down syndrome. And after the 6 months of treatment, this functional cognitivity in these networks were multiplied by 4 or 5 times. So this was a very impressive result, so promising results. So GnRH could be a cure to restore some cognitive function in some people, maybe to mobilize the cognitive reserve in some categories of patients. And we can wonder whether this could be applied to other diseases such as Alzheimer's disease, for example. And why Alzheimer's disease? For example, an Alzheimer's disease that out of 3 patients, 2 are women, so there's a sex disequilibrium here. And what do women experience in fertility? Menopause at the age of 50, and menopause means the exhaustion of the growing ovarian follicles, and thus the inability of the ovary to produce estrogens. And estrogens are the hormones that will inhibit the GnRH neurons in the brain for during 80% of the time in the menstrual cycle, and then the GnRH neurons will be active only 20%. And here at menopause, there is no more inhibition, and the GnRH neurons are constantly active for 20, 30, 40 years, as long as you live. And so they might get exhausted over time. And indeed, here there was a fantastic study performed on young menopause women and aged menopause women, and we saw that indeed in young menopause women the pulsatility of GnRH release was accelerated, but this was decreased in a 72 years old postmenopausal woman, and you had a huge drop in the quantity of LH, that is again the surrogate marker of GnRH, suggesting that in women, maybe they are more prone to develop cognitive decline and prone to develop neuropathology because of a decrease in the activity of the GnRH neurons over age. And so we tested this hypothesis in one of the mouse models of Alzheimer's disease, that is a Tau22 mouse model. And here again, when we analyzed the pulsatility in these mice, we saw that they had, like the Down syndrome mice at 9 months of age, an alteration of the amplitude of the pulses. And when we implanted this pump in this Alzheimer's model of Alzheimer's disease, mouse model of Alzheimer's disease, so at 9 months of age, these mice were unable to recognize new odors and also unable to recognize new objects. And implanting the pump rescued somehow these abilities. And here also, I want to end with a recent study just got published last Monday about COVID-19. So that's not a very good news. We saw that in the few brains of people who died from COVID-19 in 2020, that we had a viral infection in the brain, and that the GnRH neurons themselves were expressing the receptor for the virus ACE2, and that they could be infected by the virus. So out of 4 patients, we had 1 where we could see the GnRH neurons directly infected by the virus. And in vitro, we have cell lines derived from human fetuses olfactory plaques, and when we infect them with the COVID-19, we see that indeed these cells that will produce GnRH neurons can be infected. And so this was in 1 patient, but in all patients we analyzed also something that was very worrisome is that the GnRH neurons had a very strange shape and they were dying. Here you see that in this person here, you see two neurons that are expressing caspase-3, they are dying. And then analyzing so in a collaboration with a London group, we could see patients 3 months after a light COVID infection and 1 year after. And we saw that at the first visit, for example, reasonable 10% of the patients had a loss in testosterone levels that was associated to a loss in LH release. So usually when you have GnRH release, you will stimulate testosterone production, and testosterone will inhibit GnRH. So when testosterone goes down, then GnRH needs to be very high to try to compensate. So this is how it should work. So for example, like the orange one, low testosterone, higher LH. But here you have patients that are hypogonadic, have an hypogonadotropic hypogonadism, because they have low testosterone and also low LH. So it means that the brain is not functioning properly. And here, interestingly, in these patients, so that we could see in the second visit, so all of the orange and red ones, so means they had a GnRH axis altered at the first visit. They 1 year after, they were doing better. But what was very interesting also and strange is that some patients that had a normal HPG axis at 3 months after COVID injection then had a deteriorated activity of the GnRH neurons 1 year after the COVID infection. So voila, I will end here, and the most important slide, I would like to thank the people who did the work. So the work on Down syndrome was done by a very brilliant PhD student, Perry Leysen from Belgium, and a postdoctoral fellow from Spain, Maria Manfredi Lozano, and also Mikel Adamo, who conducted as a PhD student the study in patients under the direction of the Professor Nelly Pitelou at the CHUV de Lausanne, and the COVID-19 patient cohorts was analyzed by Professor Walshit Dilo at Imperial College London. And of course, I would like to thank the funders and you for your attention, and I will be very glad to answer your question if you have any. Thank you very much.
All right. Without further ado, Vincent, thank you so much for the wonderful presentation. We have a number of great questions here. Maybe one we can start with is how long does the treatment with the pulsatile GnRH last, usually after you cease infusion?
So this is a very important question that we are currently trying to address. So what Nelly Pitelou is doing, right, did already, is to, so the patients receive the pump for 6 months, and then they remove the pump, and then she receives and does the same tests in the patients 6 months after the cessation of the pump. And what happens is that a fraction of them appear to maintain their cognitive performances, and others, they drop. For the others, they drop. So, and now she's currently analyzing the imaging data to see whether this is associated with corresponding changes in functional connectivity using resting state fMRI. So we should have the answers soon. In mice, we unfortunately we did not perform these experiments, but it's something that we have to do.
Okay, fantastic. Great response. That I think leads into another question here. I think maybe on some people's minds. What are the likely or possible underlying mechanisms for the effect of GnRH on cognition?
So yeah, this is also a very important question. So the data we got from mice, both from RNA sequencing in the hippocampus, and then also the in vivo electrophysiological data, point towards an effect on myelination. So maybe you have a remyelination of some circuits that got unmyelinated or demyelinated somehow, and this could be something. And what is interesting, we also are analyzing myelination in the brain of the patients using some MRI approaches, and here there is a tendency to increase myelination in some areas. So this is, but it did not reach statistical significance because we have only 7 patients. But also another thing is that clearly we improve functional cognitivity in some networks, default networks that were known to be asleep, let's say, in Down syndrome patients. But what is also interesting, there are other neuronal network, default neuronal networks that are known to be exacerbated in Down syndrome patients, including the network between the hippocampus and the amygdala that increases anxiety. And that's why maybe the persons with Down syndrome are less performant in learning, is because they are anxious. And the GnRH treatment decreased the intensity of this hippocampal amygdal functional network. So depending maybe on the neuronal populations that receive the GnRH information, maybe the action of GnRH will be different, but as a whole, it could improve the ability of the person to perform cognitive tasks.
Oh, incredible. Do they report decreased anxiety as well then?
Yes, it's something we have measured, and indeed they are less anxious.
Fantastic. So I suppose if there is a role for demyelination, this perhaps also can explain why it may play a role in a number of different conditions: Down syndrome, Alzheimer's, COVID, etc.
Yes.
I'm not sure.
Sorry.
I was just going to say, I'm not sure if you mentioned this, but is that is there an association with COVID and brain fog then? Is that a possible connection there?
Yes, exactly. So it's what we were looking at, and for sure. So here, the British cohort we were following had only 47 patients. That was reasonable, but too low to reach statistical significance. But indeed, there was a trend to have a more impaired memory and neurological symptoms in people who had a defective gonadal axis versus the others. But then it's just a trend, it's nothing significant. We should do the study on a few hundreds people to see whether it's indeed the case. But it's what we suppose. If you have your neurons that are less active or less fit in this population subsequent to the infection, maybe it could contribute to the fog and the alteration of cognitive functions.
Incredible. Okay. Somewhat along these lines then as well. So in the Down syndrome studies, these were adults that were being treated. Is that correct?
Yes.
So the question here is, could you also intervene earlier in life, in childhood, for example? Would it be more efficient, and would it be even protective, I suppose?
Yeah, so this is another important question that we receive a lot from people, families who have relatives who carry the Down syndrome. So here, clearly, we cannot intervene before puberty onset. So, because if we do intervene before the age of 15, for example, we would do more bad than good, because we will interfere with this important process. So the idea here would be to intervene after puberty, that is to say, around the age of 15, maybe. And this, yes, we could try to do it as soon as 15 years of age. Or also what we could, and this is what we are exploring currently. For example, in the mouse model, as you said, what we found in this paper is also that the origin of the alteration of GnRH production was at minipuberty. That is to say, this period that is between the first week of life and 6 months of life in humans. And here in Down syndrome mice, apparently the GnRH system is more active. So minipuberty is a window in which we could intervene, and either to decrease the amplitude of minipuberty, or if it's not present, or if minipuberty is not present, indeed here we could give GnRH to children to mimic correct minipuberty. And here we have another study showing that if you correct the amplitude of minipuberty, you change neurodevelopment, and you can improve cognition for the whole their life. So, well, this is another window of opportunities for intervention. It's minipuberty, and it's something we are exploring both in mice and babies.
So I suppose then effectively you can be protective in terms of cognition, but there are other considerations in early childhood, etc., in terms of development and normal development.
Yes, so but minipuberty, apparently correct minipuberty is key. And for example, you have a population that is babies that are born prematurely, premature babies. These babies are known to have a higher amplitude of minipuberty, very high minipuberty levels of gonadotrophins at minipuberty. For example, to give you an example, a girl with what is premature girl has FSH levels that are 200 times higher than a girl born at term. And here, interestingly, we showed that to correct the amplitude of minipuberty, you can give nitric oxide, that is an inhibitory molecule on the activity of the GnRH neuron and the expression of GnRH. And fortunately, and we discovered that by speaking with medical doctors, in fact, nitric oxide, inhaled nitric oxide is a cure, a therapy that is given to prematurely born infants to save their life, to mature their lungs and open their airways. So this molecule is already given to children. So here we are exploring whether if we prolong it's when they give it for a longer time, whether they correct also minipuberty and thus improve later on their neurodevelopment and their learning abilities, for example.
Incredible. Yeah, it's fascinating stuff. Thank you very much. This question ties in somewhat to some of the earlier ones. You mentioned that the treatment is well tolerated. Are there any notable side effects? You focused on cognition here, I suppose.
No, so because you give the natural molecule and the natural rhythm, you have no side effect except maybe the irritation on your arm. However, you have other therapies that are given in children, for example, to suppress puberty in children with precocious puberty, or in transgender children to stop their sexual maturation. You give either agonist or antagonist. So it's a weird system, because both agonist or antagonist, because the agonist act longer on the receptor and the antagonist inhibit it, both internalize the receptor and have an inhibitory effect. And for example, and this could have deleterious consequences on cognition. And for example, men with prostate cancer are treated with this GnRH agonist to suppress testosterone, to suppress the axis. And it's very well documented that they experience cognitive loss, and even they have higher risks to develop Alzheimer's disease. Before our study showing that GnRH could have an effect on cognition, it was not understood why. So here we have, so this reinforces what we have found, because indeed it's clear, it's documented, these patients develop cognitive decline. And it's the same for women who have endometriosis. For example, they are treated also with this GnRH agonist, and they also experience cognitive loss, and they may also have higher risk to develop Alzheimer's disease.
Wow, incredible. It seems to be quite wide-reaching then, the potential implications of your work. A great question here. Thanks for the wonderful presentation. Agreed. In GnRH neurons projections, can iDisco data indicate the presence of GnRH neurons between the hypothalamus and hippocampus?
So other cell bodies. So indeed, so what we did a very interesting experiment is that to inject a retrograde virus in the median eminence, the projection site of the neurons that release to talk to the pituitary. And this retrograde tracing showed that most of the fibers that are reaching the hippocampus and the cortex in mice are from the same neurons that control reproduction. So this is quite intriguing. But indeed, you may find on the way some cell bodies of GnRH neurons outside the hypothalamus, and this is even more important in humans because in mice we have about 800 neurons between the olfactory bulb and the hypothalamus. In humans, you have 2,000, but you have 10,000 neurons that also migrate outside of the hypothalamus, at least at the fetal level. So we don't know if after birth these guys still express GnRH, but indeed they migrate from nose to the hypothalamus and also to the cortex and hippocampus in humans.
Okay, excellent, great. Do you think GnRH pulse injection is working in menopause or overrectomized mice, as in minipuberty?
So this is also a fantastic question. So also, unfortunately, it is known that women at the transition of menopause, they experience cognitive loss. So fortunately, they recover this cognitive loss at a distance of the transition. But then there is an American study that was done on 2,000 women showing that, so they showed this recovering of cognitive loss after the transition, but then unfortunately they analyzed the same woman 4 years at a distance of 4 years, and they saw that the cognitive performances 4 years after menopause of the same woman were worsened when compared to the one before menopause. So indeed, menopause itself could induce cognitive alteration problems due to this wrong exacerbated pattern of GnRH release. So indeed, maybe the GnRH pump could be useful also in this for this application in women at menopause, and it would be even maybe safer than giving combination of steroids or something like that, because here it just acts that acts on a single receptor in neurons.
Okay, fantastic. I think we can probably squeeze in two more here. A simple technical question: how do you implant the pumps, and do you have to re-implant during the studies that you described, or are you just preloading them with the correct amount of?
So here, as Tseng told us, we started very soon when it was on the market, and we used both the first generation and the second generation. So what is really cool is that you can refill the pump. So, and it's the same in patients. You have to change the pump every 3 days. In mice, fortunately, it lasts a bit more. For the reservoir, I think you have to fill it within the 15 days 2 or 3 times. But the limitation here is the power, the battery. And the first generation, and depending also on the changes you ask to the pump, it's more or less demanding in energy. And so the first pumps, we could, for the most, do two weeks of experiments. So that's why we did the pump during 2 weeks only. But then the new generation, now we could do it for 50 days. And then, of course, you have to refill through the skin, but then you can use them for 50 days. So it's a great improvement.
Okay,
It's,
fantastic. I think we will try and squeeze one last one in. With the Down syndrome studies you described, I think you mentioned they were male patients. I think if I recall correctly, it's predominantly male, seen in males Down syndrome. Are there sex differences
So
in effects?
this is another great question. So initially we were not intending to do this study only in men, but for women the inclusion criteria are a bit different and more difficult because they need to not take the pill, because if not, it would interfere with the treatment. And in addition, they need to be received for the test at a specific moment of the menstrual cycle for the hormonal condition not to interfere with MRI, functional cognitivity, for example. So it's more stringent, but the new assay that Nelly Pitero will be developing, she will do it double blind with a placebo arm in 30 men and 30 women, so for sure. So, but voilà, here it was just by let's say chance. But she got 8. So we in total now she has 8 patients, and the last patient was a woman, and she had the same improvements than the men.
Okay. Fantastic. Unfortunately, I think that is all we have time for today, Vincent. But I just wanted to say a huge thank you for presenting your research and sharing insights with us today. It's been a pleasure having you with us.
A shared pleasure. Thank you so much.
Okay, thank you.
Bye. Bye, Andrew. Bye, Chen.
Bye. Another huge thank you to our partners and today's sponsor, PrimeTech, the makers of iPressio microinfusion pumps, for helping make this series and today's event possible. And in closing, we hope you enjoyed this Inside Scientific webinar, and we hope to see you again next time. Thank you, everyone.